and we have the promising current role of gene and cell therapy as a treatment for neurological disease. But before I make some brief introductory remark, why don't we have the panel introduce themselves, please?
Okay. I'm Steve Paul, CEO of Voyager Therapeutics, formerly EVP of Science and Technology at Lilly, and formerly Scientific Director of NIMH.
Hello, I'm Gillian Leng. I'm the Deputy Chief Executive at NICE, and in case there's someone who doesn't know, NICE looks at new drugs and looks at their cost effectiveness and decides whether or not they can be funded in our health service.
I'm Tim Cox, and a physician and researcher, and work now on gene therapy for a very rare disease and other single gene disorders, which actually are susceptible to therapy in a rather wonderful way.
I'm Brad Hyman, a neurologist at Mass General in Boston, and I direct the Alzheimer Center there.
Right, to set the stage from where we were earlier this morning, it is definitely about innovation here today. And this panel will show us that new innovation in technology also affects the way we potentially treat and help the patients. So cell and gene therapy are newcomers to the scene, relatively speaking. And this creates a very interesting conundrum for us. Certainly in pharmacology, we're used to deliver molecules, drugs, and we have a whole pathway to the patients, including regulatory approvals, that's been shaped by the modality in some way. The chemistry and the safety is based on prior experience. Along comes the revolution in biotechnology, exemplified by a number of companies who did recombinant protein therapy. And they now created a new demand for that process to work better. And eventually, along comes discoveries driven partly by the understanding of genetics, but also just as a new modality of delivering a protein through gene therapy. And on top of that, to make life even more complex for all of us, actually patients can be helped by cell therapy. And certainly all of that taken together creates both opportunities and challenges. And for example, even though our panel and I will discuss mainly gene therapy with you today, it is certainly true that cell therapy in the medical world is making an entrance. Certainly CAR T therapy, chimeric receptors on T cell was not thought of until very recently. And in the stem cell world, where there are many clinical trials, most of them outside the central nervous system, but there is a concerted and very sophisticated effort to generate cell types in the same way as we develop molecules and drugs to treat very specific syndromes, including Parkinson's disease. But today, let's start with the here and now. And Steve, you've been working in the industry for a long time. What do you see as the relevance and the potential impact of gene therapy?
Okay. So why gene therapy and sort of why now? And this is a field that, like many in medicine, have had a bit of a rocky course over the past 30 years with starts and stops and controversies, et cetera. But I think there are three main drivers of why there's been a resurgence of interest in gene therapy. One is that we've identified mutations that cause a number of, in our case, inherited neurological disorders. These are monogenic disorders or recessive disorders. The mutations are unequivocal. Two, there's been a lot of work done on the biology of these diseases. How do these mutations, either loss of function or gain of function mutations, cause the disease? And then three, we have what we think is a relatively safe way of delivering genes or silencing genes in the central nervous system. And this is using a little virus that we sort of use to get genes into the brain. It's an adeno-associated virus. And we literally use it as a vector, a way of getting drugs, in quotes, the genetic payload, into the brain. And so I'd say the resurgence of this has been going on maybe the last decade or so. And we're now getting into the clinic, we, the field, collectively. And we're starting to see some rather remarkable clinical benefits. So spinal muscular atrophy, single injections in SMA1 look to be very effective. It's still early days. At our work at Voyager, we're putting a gene that allows the conversion of the medicine levodopa to dopamine directly into the basal ganglia of patients with very advanced Parkinson's disease to get them now to respond better to their medicine. In systemic diseases, we're seeing some remarkable effects in hemophilia with one-time injections of factor 9 or factor 8 to the liver. And there are some inherited retinal diseases that cause blindness with some fairly dramatic effects of late. Still early days, but most believe that we can now reduce this to practice. We're also encouraged by the fact that the little viruses that we use to get these genes in the brain or silence these genes are getting better and better. We now have capsids that get into the brain across the blood-brain barrier 100 to 200 times better than the current crop of capsids, which augurs well for getting broad brain distribution for diseases like Alzheimer's disease. So it's an exciting time. To me, it's a bit reminiscent of the early days of monoclonal antibodies before we could fully humanize them. There were fits and starts, and now, I think 8 of the top 10 selling drugs in the world are now human monoclonal antibodies. So it's an exciting time for gene therapy.
Thank you, Steve. So in this context, then, we understand gene therapy, as you say, monogenic diseases. We also, of course, have Brad Hyman on the panel here, who has many contributions to the field, in particular in Alzheimer's disease. So we're curious to hear from you what your perspective is, gene therapy vis-a-vis more common diseases like Alzheimer's disease.
Well, I think I agree with Steve completely that it's an incredibly exciting technology that really feels like it's maturing and about ready to burst onto the scene. Without doubt, the retinal degenerations, the lysosomal storage diseases, and so forth are the sort of low-hanging fruit there, because it's essentially equivalent to replacement therapy that we're familiar with and have a kind of medical model for. I think then sporadic Alzheimer's disease or disease like that, echoing part of what we heard earlier today, understanding the underlying biology is going to be critical to figure out how to impact it. But there are 2 examples that I think really already stand out. One is the development of a series of resilience genes, and my personal favorite is ApoE2. I know Steve has worked on this for years as well, where inheritance of ApoE2 is profoundly protective in the human population against Alzheimer's disease, and a really an unproven hypothesis, but a hypothesis would be that if you could express ApoE2 in the central nervous system, would that be protective as the disease goes on? And there's arguments in favor or against, but the technology to do that experiment, I think, is evolving. And the second possibility, I think, is, again, to echo something that I know Steve is deeply involved in, is to sort of combine these notions of monoclonal antibody and gene therapy, and in recent work generating an antibody that's directly expressed essentially in the brain. And this could also be in a cell therapy, any sort of secreted molecule. That might be a way to overcome the blood-brain barrier issues, which have really, been very, very challenging to tackle using sort of standard immunotherapy approaches. So I think those are the 2 places sort of that there may be direct application in a disease that doesn't have a clear autosomal dominant inheritance, resilience genes, and then the ability to express secreted proteins in the CNS to overcome blood-brain barrier issues.
Right, so to come back to where we have some experience, Tim Cox, the rare lysosomal disorders or the rare diseases in general present us with this opportunity that Steve mentioned of monogenic disease, maybe a very precise needs, and also a very specific population that where we know that the physiology and the pathobiology and the genetics actually link up. Yes. What is your perspective on these new technologies?
Yes, it was very nice of you to mention lysosomal diseases, because of course for those of us who are enthusiasts and have the pleasure of seeing the field over so many years develop from its discovery in the 50s, you really do see now that the lysosome has a number of extraordinary properties. It's at the center of the cell. It's not just a recycling organelle, as we tend to think of it in the earlier days, but it's involved in energy sensing, in metabolic control. It's highly adaptive and indeed interacts with so many of the key regulatory factors in the cell. And the discoverer, Deduve, realized very early on that it was accessible. So you could complement a defective lysosome, if there was just a unitary defect in it, at a distance by providing the missing protein, if you will, the missing factor, in a whole field from a small place. And indeed, the studies by my colleagues have shown, actually, in a really horrible and complicated disease, it's possible to correct from a small factory in a small silent area of the brain, if you will. Sorry to mention that to the neurologist. It's possible to get a wild field of correction right throughout the whole neuraxis, and that's been very astonishing for us to see, with a continued production, for, as we know in dogs, up to years and years, 10 years, from at least from a complementing virus. Now, as a former jobbing clinician and now in the field of interest that I've always cherished, my personal view has always been treasuring your exceptions, the insights you get from the very rare to everything else, because, of course, the whole thing is interrelated. And the one example, which is well known, no doubt, to the panel, but perhaps not to everybody, and there are many others, is, let's take Parkinson's disease, right, or Parkinsonism. So there's a condition, huge genome-wide, GWAS studies, I won't tell you how many tens of millions of studies, completely missed that the greatest genetic determinant for Parkinson's disease in all populations is a mutation in a gene, GBA1, which is mutated in Gaucher disease, a lysosomal disorder. You only have to be a heterozygote. You don't have to be a homozygote to get Parkinson's disease with a 5 to 10-fold increased risk. It's a huge risk, not a tiny percentage at P0.01 with 100,000 people. It's the insights that the lysosome, it turns out there are other lysosomal disorders less prominent in the general range than Gaucher. It's a disease in part of the lysosome. And of course, since we can access Gaucher by various means, the recombinant enzymes, a substrate-reducing drug that would enter the brain, which is in development, in clinical development now with Sanofi's enzyme, and indeed other approaches to reinforce or preserve, we have the opportunity to do some really big things, some proper clinical experiments with definable effects. So the rare as an insight into the new and the common, and the economy of scale, if you will, of the science, but also the astonishing door, the astonishing little letterbox that the lysosomal system offers, central to cell metabolism, turnover, and cleaning things out.
Thank you. So, but now we understand that some of these new technologies also present opportunities of doing a one-off, as I've learned in my English dictum this morning, one-off therapies or one-time only therapies.
One and done. One and done.
One and done.
Yes.
One and done. There you go. That's our way of saying in America that, we fixed the problem. Now, Gillian, so when we come up in that scenario, again, our current way of dealing with reimbursements, our current way of dealing with payments kind of break down, because now you have to calculate in some way that it's certainly worth for the patients to get this therapy, that we know. But how do you actually, in your role, how do you balance these things? And is there any way we can think clearly about this to not impede the access of these drugs to patients?
Good question. And NICE has been doing a lot of thinking over the past year or so around cell and gene therapies and whether or not they are indeed appropriate for us to look at using the methodology that we have. And a report was commissioned by York University to answer that question and to put in some hypothetical examples. Some of you might have read the report from York. It's on the NICE website. And the conclusion was that NICE's methods were fit for purpose and we should look at these therapies. And it's good because NICE has an important role to drive the uptake of innovative medicines in our health system. But of course, we've got to protect the public purse while we do that. So we can't ignore the costs of the treatments alongside the long-term, lifelong benefits that they might have. And in terms of how we go about that, There are challenges in relation to the evidence base. It's likely to be relatively short term. The studies are going to be single-arm studies, and the cost is interesting. A colleague of mine who's more involved in this on a day-to-day basis than I am, before I came here, he said, the pricing models are bizarre, he said. And we need to understand the approach around pricing. We want to get treatments to patients. We want to make sure there's a sustainable funding model. So we've got to have a dialogue with industry to work this through. Some of you might have seen the recent consultation that we had looking at how we manage our highly specialist technologies program for the future. It's been running over the past few months, and it's likely that things currently in the pipeline that are gene therapies will fit into that program. So we hope that the way that's been described, with a much higher cut-off cost per QALY than our standard program, and with QALY weighting, will help. But the conclusion has been that the standard NICE approach is appropriate.
Steve, so how does that compare to the rest of the world? The pricing models that you have to develop in a insurance model country versus this government-based reimbursement model?
Well, I think the payers to date, at least in the US, have been fairly receptive to what I'll call sort of high-priced treatments for relatively rare diseases, in part due to the fact that they're relatively rare, and so the aggregate costs are quite modest. So I think for things like we're working on ALS, a subgroup of ALS patients that's due to a mutation in SOD1, and we're silencing that mutation. We'll be in the clinic at the end of this year. There are only 600 people prevalent with that mutation in the US. It's a bad, bad disease. It has a median survival of about 18 months, which means half the people that have that mutation are dead at 18 months and the rest are dead. So if we prolong survival by 2 or 3, 4 years, or prevent people from having to go on a ventilator or respirator, what would that be worth? It's going to be, it's cost us a lot of money. It's going to be not a cheap treatment, but I think the prevalent population is pretty low. I think where this gets really complex is when you start talking about a larger population. Even, as you know, the PCSK9 antibodies, which are monoclonal antibodies that block PCSK9 interacting with LDL receptors, that's now you're getting into a much more common market of, hypercholesterolemia. That's where the payer system is sort of balking and saying, well, why don't you try 10 statins before you get there, kind of thing, and balking on price. So I think where this is going to get interesting is if Brad is successful with delivering ApoE2 or silencing ApoE to treat Alzheimer's, then it's going to get to be a very interesting and taxing sort of problem and dilemma. I also believe that this is an opportunity to introduce pay for performance. So if you have a gene therapy that really does effectively treat a disease, maybe over time there are payments made by payers. If patients don't respond, they succumb to their disease, let's say in 6 months or a year, maybe you don't pay. And so there are some opportunities here, I think, to get creative about reimbursement.
Right. And just a comment, since I guess I'm only representing the cell therapy on the panel, is that I remember when the cell therapy for Parkinson's disease was developed, one of the things that struck us was the interest from the developing world. Because even as we think of L-DOPA as a substitute for the symptomatic loss of dopamine, they did not have access to the $6,000 to $10,000 a year that was per patient. No. So the one-off, once you're done treatments were very interesting to them. So we can certainly imagine with very advanced way of making GMP products, cells, that replace, when these drugs actually fail, partly for physiological reasons that you get side effects because drugs are not feedback controlled. If a cell can do that job and do it permanently, it actually becomes, as you point out, more economical and more accessible over time. So maybe the pricing model in an odd way, particularly with this idea of that you have an outcome base so that if your cells work, then the system pays. It could be, yes, Tim.
I was just going to say, there is an example with Gaucher disease, which has a high-cost enzyme therapy. And in the developed world, I'm not referring to just to South Korea, but also in the semi-developed world, Pakistan, actually, where this disease occurs very severely in children, in one caste of peoples it's referred to. There, actually, it is bone marrow transplantation, which is curative, but of course carries a mortality, is actually coming back, because you could get a philanthropic donor who might give for a year or two and have his newspaper image and so on and do very well for a year or two, but for a lifetime of therapy, then no one is going to pay the exorbitant costs of enzyme therapy from any supplier. And so marrow transplantation, with the attendant risks of simple marrow transplant, which is a cell therapy, is now actually being reorganized in Lahore, for example, for this wonderful professor's patients. So it's a kind of retrogressive step, but it does show the value of a one-off therapy where you've got total efficacy.
Very interesting. You have a comment about that?
Well, I was just going to comment that in the new approach, we very much want to recognise the potentially increased length of life and quality of life, which could be huge compared with the other types of, say, cancer treatments that we currently look at, hence the QALY weighting, which might take the threshold up to £300,000 per QALY, vastly more than for the other treatments that we're currently approving. And we really would like to engage in relation to what we might describe as innovative pricing models. The one that we're using at the moment relates to the Cancer Drugs Fund, where things that look promising but don't have enough evidence will be funded for a defined period to collect the data. That's one model that we've got in the UK at the moment, but very keen to engage in relation to other proposals.
Yeah. One of the challenges there is that you measure efficacy often as a mean or a median in a large patient population. That's how drugs kind of get approved versus placebo. But when you look into those populations, you often see a group of patients that respond remarkably well. We saw this a lot with many of our psychiatric drugs at Lilly. You see a group that responds, but not as well, and then you see a group that doesn't respond very well. And so you at NICE say, okay, well, how effective is this drug? Well, it's effective based on the median. Often, and cancer drugs, quite frankly, fall into the same category. A very modest percentage of people, will respond very well to a cancer drug, and most patients, many patients, with some exceptions, obviously. So I think one has to be mindful of the fact that, there are subgroups of patients that can really benefit on these drugs.
Absolutely, and we will always look for subgroups, data permitting, and I think about 15% of our recommendations are for subgroups of the licensed population.
Very good. All right. So ultimately, I believe the best research and the best medicine is always done in a patient-centric manner. And that is one of the reasons we can all be together here in this unity of purpose. And so, Brad, I remember talking to you about this many times, but in the practice that you are doing with patients, you frequently have to envision the step-by-step approach to the patient, like we just saw this morning, to you making a lumbar puncture, I remember watching you do, and then bringing these new therapies to the patients. What do you see, or what do you imagine when you hear this discussion about how neurological practice may change, because we've typically been quite insular in the way we deliver drugs? Will you collaborate with immunologists, neurosurgeons, etc.?
So I think that's a really interesting sort of vision of the future. How do we recommit our medical practices, both at academic hospitals, and then depending on how you look at it, either more easily or less easily in the sort of real world, in the developed world, and then, as you point out, in the developing world. And I think there's going to be some real challenges there. Certainly talking to my patients about a variety of research projects, there's some hesitancy, even in Boston, even a very well-educated community, to do something that's experimental. And the closer you get away, the further you get away from a pill, the more resistance there is from the patients. And I don't know if you have experience. I imagine that in a autosomal dominant disease where people will have some understanding that it runs in the family and that genes have a more personal meaning to them, that you might be able to have a little bit better ability to communicate. But in a sporadic disease, especially in elderly individuals, I won't be surprised if we find a tough time recruiting for these trials.
Well, I can actually comment on that. Surprisingly, Harvard Stem Cell Institute did surveys when they started about 12 years ago about the way patients would look at cell therapy. And they were surprised to find more of this kind of reasoning, that if it really will take care of my disease, I'm willing to do this. Of course, with the psychological risk of desperation, but in a very organized manner, with the sense that the FDA, the EMA, and all the classic ways of looking at safety and efficacy as a combined effort, their findings were that people were not so hesitant.
I think that's right. If there's a therapy that is established in some sense, that there will be an easier way to have the patients accept it. But
if it's incremental.
But it's the incremental trial, which is going to be going to take some gentle persuasion.
Right. Okay, so in the last 10 minutes or so, we have an opportunity for you to look to the future. Steve, this is what you do on a daily basis, I understand, but what do you see as the main challenges? Because we want to be quite realistic about what we test and solve.
Yeah, I think for a small startup company, we're very mindful of the fact that we need to still convince people that what we're working on is likely to work. And so we've stayed pretty focused, again, on these monogenic disorders. Friedreich's ataxia, which is a recessive disorder, we know the mutation. We know where we have to deliver the gene. It's a gene called frataxin. Huntington's disease, we know we have to silence that gene. We know where we have to silence it. ALS, I mentioned earlier, and this therapeutic protein strategy in Parkinson's. So for us, I think we're going to be very encouraged about going after diseases that affect a broader range of brain regions once we kind of can focus and get some results in some of these disorders. But I'm pretty encouraged, not just by our work, but by the field's work. I think we're seeing success in a number of the diseases I mentioned earlier. Some of the work that Al Sandrock mentioned on antisense oligonucleotides and intrathecal administration, also very, very encouraging that gene therapy, broadly defined, is ready to be real at this point.
Very interesting. So Gillian, we had a question from the audience here that's very specific. For NICE, do QALY cutoffs get derived from a fixed national budget for therapeutics? And how do budgets compare versus those of other countries? Maybe you can define QALY cutoffs for us, not here in Britain.
Okay. We use an approach when we're evaluating drugs to work out incremental cost effectiveness. We calculate the cost per QALY, and generally speaking, we approve drugs in our normal programs that are below about 20 to 30,000 pounds per QALY. And that threshold has not been set by the government, it's not been set by NHS England, it's been the consideration over the years of our advisory committees and has been tested in consultation. And actually it's been tested quite a lot by various groups of economists and others considering that. And there are one or two people who would argue that that's too high for the amount we spend on our healthcare, that it should be much lower. There's also the drive to support innovation and uptake of new technologies too. So it's a really difficult balance, and there's probably no right answer. And so that's the sort of standard approach. So you heard me say just now about the drugs for rare conditions and the much, much higher threshold. And so what's that based on, you may ask? It's based on experience of things that we've evaluated over the past few years that fall into that category of very rare conditions. It's not an exact science. And one of the things to leave people with is that we're going to go through the process that we've just consulted on, that I've just described very briefly, but we absolutely will review it because we need to get it right. We need to get it right first and foremost for the patients that will benefit, but we need to get it right for our health economy, and we need to get it right so we're not stifling innovation. So it's a complicated equation.
On that term, so I want to get back to the opportunity for you to provide something, not just as an answer, but maybe as an opportunity for you. We're about innovation, right? So what do you see in your world as a way to facilitate these new therapies? Is there some way, you may have mentioned already, but something that you look for in the next 4 or 5 years, we certainly have time because we need to do the research, that you can anticipate maybe and provide a facilitating speed up of both the regulatory process but also the reimbursement process to stimulate our work and effort?
Well, we are very sensitive to upstream developments. There's a couple of things that we do in relation to that. One is horizon scanning. We run a PharmaScan database where there's lots of information about things that are coming forward. So we're aware of what's in the pipeline. We also engage with industry as much as industry wants to engage with us. We like to say our door is open. We have something called the Office of Market Access, and we're very keen to talk about new technologies as soon as we can and to explore what companies should be thinking about in terms of NICE's approach to evaluation. So that's understood as early as possible. And as I say, we are constantly evaluating the way we go about that, but we are keen to involve and to have conversations as soon as possible. We also link very closely with the MHRA, the UK's medicines regulatory body too, so that we're working alongside them as they're licensing new products.
So Tim, what do you see on the horizon?
Yes.
The most exciting opportunities?
Yeah, well, there's a big problem on the horizon, but if I could just chip in, just to stir it up a bit, because I think Gillian's spoken so wonderfully, but I have to say, for those who've been in a long time, the QALY cost, which is completely arbitrary, as she pointed out, owes its origin to the cost of dialysis per year, which was sorted out eventually in the 1980s. That's the actual origin, slightly inflation related, which is extraordinary. And the other thing I would say is, of course, politicians always tell us that there's a fixed budget for health, but if you really look in countries, certainly in the West and certainly the great United States, there's never been a fixed budget for health. It's a way of deputing some level of control quite properly to a very good and appointed body, but in the end, it's a societal matter, and it always has been, I would say. Forgive me for saying. If you will, very political statement.
We'll spend more money on healthcare. We agree with you.
We always have, but of course, there's a fixed budget for health is the common mantra. 10 seconds to say what I think the future is. Well, the gene therapies will come in for the clinical experiments that are going to be dramatically successful, and when they are successful, like hemodialysis and transplantation, they will get adopted because the society won't tolerate it if they're not. That would be my first prediction. My second prediction, looking for, would be, certainly in certain countries, is to remove the ridiculous conservatism about access and about screening. Care on ensuring that screening is clinically articulate for a disease which you can interrogate and interdict will be a crucial thing, in my view. We don't have it enough, but as was pointed out in every session with your particular disease, with your particular diseases, there's no disease in the book where therapy, biologically given earlier, is not going to be better. So when you've got a few certainties from the screening and greater introduction in an ordered way, then we'll deal with some of the economical scale problems. So I think the future has to deal with that, the marriage of screening, utility, and a clinically articulate outcome.
I think this point is particularly relevant to gene therapy. We're really conceptualizing going into patients who are already symptomatic with Huntington's or with mutant SOD1 ALS, but it's very, very clear that that's just the beginning. And that poses a problem is, how do you actually, use these treatments? Most of these patients know that their affected parent had the disease and died this horrible death.
Yes.
Patients that we bring into our company, all the time, very inspiring people with stories, but they wanted treatment now. They're not going to get it initially, but eventually that's where you want to correct the gene, obviously, before it manifests itself at all at that point.
We're just sticking to children at the moment, so it's easier. But yeah.
So Brad, do you have a minute of wishes on your list?
Wishes. Well, I'm going to echo much of this. Is that there's no doubt that pre-symptomatic therapy is going to be a better way to save the brain, to prevent organ failure, than trying to sort of fix things downstream. And yet there's going to have to really be a paradigm shift, especially for experimental therapies like gene therapy, in order to develop both the technologies to have certainty about that future and also delivery methods. And so I think we talked earlier today about collaborating with engineers and with physicists and so forth. That's not just the nuts and bolts of how do you make a boatload of virus, because you're going to need an awful lot of virus. It's also how do you get the technologies that will allow pre-symptomatic diagnosis to a point where it can be screened for by the general practitioners. And those are real challenges, but it's also exciting to at least be able to visualize that future.
Well, just perhaps to round up and again provide an opportunity, a slightly different version of that reality could be that also for restorative therapy, at least for the few that may be restorative in a symptomatic way, like Parkinson's disease, and maybe some of the other diseases, if you have a glial cell replacement that's non-immunogenic in MS and so on and so forth, could also work the whole spectrum of the healthcare system. But I think overall here we have the opportunity of using innovation in technology that drives solutions for the patients and presents completely new problems to solve at every level, from the lab bench to the regulatory and the payment system. So I would like to thank all of you and thank the audience for listening.
Thank you, Ollie.